This split-screen counter tracks the sheer volume of human gamete production. On the male side, a single instance of ejaculation releases an estimated 200 to 300 million microscopic cells. Contrast that against the female yield over a lifetime. From an initial prenatal pool of 2 million, the reserves drop to roughly 60,000 by puberty, ultimately outputting exactly one viable cell per ovulatory cycle. The production of these gametes, sperm and ovum, is called gametogenesis. It occurs within the primary sex organs, the testes in males and the ovaries in females. Both systems face the exact same biological prerequisite. A standard human somatic cell contains a diploid count of 46 chromosomes. To create a viable embryo, both the male and female gamete must reduce that number by exactly half, down to a haploid state of 23. To execute this chromosomal reduction, both biological sexes rely on the exact same universal mechanism, cellular division by meiosis. Yet, despite relying on the identical meiotic foundation to reach 23 chromosomes, the male and female systems apply it in radically opposite ways. They execute entirely divergent strategies in how they manage their biological timelines, their cellular symmetry, and the allocation of physical resources. The male reproductive strategy is built strictly for continuous, high-volume manufacturing. This process occurs deep within the inside walls of the seminiferous tubules, operating as a continuous biological assembly line that powers on at puberty. This cellular lineage tree maps the initial multiplication phase. The line begins with a single immature spermatogonium. This diploid cell holds 46 chromosomes, multiplying continuously through standard mitotic division to ensure a steady supply of raw material. Periodically, one of these cells, now classified as a primary spermatocyte, enters meiosis I. It undergoes a reduction division to create two perfectly equal secondary spermatocytes. Because of the meiotic split, both of these new cells are haploid, possessing exactly 23 chromosomes. These secondary spermatocytes rapidly divide a second time to complete the meiotic cycle. This final split yields a perfectly symmetrical row of 4 identical haploid spermatids. The physical result is a massive, relentless current of microscopic cells, tightly packed and continuously pushed forward through the enclosed duct system. The arithmetic of this biological factory is entirely rigid. Every single primary spermatocyte that enters the line produces exactly 4 equal gametes. This strategy is aggressively optimized for geometric multiplication. Spermatogenesis prioritizes sheer quantity and rapid mobility over individual cell durability. This entire manufacturing process remains completely dormant throughout childhood. It requires a specific trigger, a sudden, significant increase in gonadotropin-releasing hormone, or GnRH, secreted by the hypothalamus at the onset of puberty. This anatomical flowchart tracks the resulting endocrine cascade. When increased levels of GnRH act on the anterior pituitary gland, they stimulate the secretion of 2 distinct gonadotropins, luteinizing hormone and follicle-stimulating hormone. These signals are highly localized. LH acts externally on Leydig cells to stimulate androgen synthesis. Simultaneously, FSH acts directly on the Sertoli cells within the tubule, secreting factors required for the final stage of maturation. That final stage is spermiogenesis. It is the physical teardown and rebuilding of the cell, transforming inert, round spermatids into highly specialized spermatozoa. The architecture is stripped to its absolute minimum. The elongated head contains the haploid nucleus, capped by an enzyme-filled acrosome to breach the ovum. Directly behind it, a compact middle piece is heavily packed with mitochondria. Those mitochondria generate the intense kinetic energy required to violently whip the tail, providing the motility essential for fertilization. Once the physical transformation of spermiogenesis is complete, the heads detach from the Sertoli cells.
This physical release into the seminiferous tubules is called spermiation. From there, the gametes travel through the accessory ducts. Secretions from the epidermis, vas deferens, seminal vesicle, and prostate mix with the cells to constitute semen, finalizing their capacity for vigorous motility. Guided by a precise hormonal cascade, the male reproductive system engineers a standard cell down to the leanest possible genetic delivery vehicle, systematically eliminating any internal biological mass that does not contribute to speed. Oogenesis, the formation of a mature female gamete, abandons the continuous assembly line model entirely. Its timeline is heavily front-loaded. This timeline mapping female cell count reveals that the entire process initiates during early embryonic development. Millions of gamete mother cells, or oogonia, are generated within the fetal ovary. After birth, no new oogonia are ever formed. These fetal cells immediately begin meiotic division, but long before completing it, they hit an enforced biological pause. They enter prophase 1 and arrest there temporarily, becoming primary oocytes. While suspended, each primary oocyte is encased in granulosa cells, establishing a primary follicle. From birth to puberty, millions of these follicles degenerate. The original reserve is reduced to just 60 to 80,000 follicles per ovary. The female system is not a factory. It is a strictly finite, rapidly depleting vault of arrested cells, subjected to aggressive biological culling decades before they are ever required to function. At puberty, a select few of these surviving primary follicles resume their development. They acquire additional layers of granulosa cells and a new boundary layer called the theca to become secondary follicles. This cross-section of a maturing follicle illustrates the transition to the tertiary stage. This phase is characterized by the appearance of a fluid-filled cavity called the antrum and the rigid organization of the theca into distinct internal and external layers. Within this tertiary environment, the primary oocyte awakens from meiotic arrest, completing its first meiotic division. Unlike the perfectly symmetrical split in male gametogenesis, this division is severely unequal. The parent cell pushes nearly all its nutrient-rich cytoplasm into one side. The result is one massive haploid cell, the secondary oocyte, and a tiny discarded satellite cell known as the first polar body. This stark cellular asymmetry provides a crucial advantage. By hoarding cytoplasm, the secondary oocyte guarantees the intense nutrient reserves required to sustain itself during the earliest stages of post-fertilization development. The secondary oocyte then secretes a new surrounding membrane, the zona pellucida, while the surrounding structure finishes its transition into a mature Graafian follicle. Desequence concludes when the Graafian follicle forcefully ruptures, ejecting the large secondary oocyte from the ovary in the process of ovulation. The female meiotic strategy deliberately forces an asymmetric cellular division. It routinely sacrifices volume, culling millions of its own cells to perfect the viability and resource consolidation of a single ovum. This side-by-side comparative table maps the completed processes. Plotted together, the divergence of gametogenesis becomes entirely clear. Spermatogenesis operates as a high-volume, egalitarian machine. It runs continuously, symmetrically splitting one primary cell into 4 highly mobile genetic delivery systems. Oogenesis operates as a front-loaded asymmetric vault. It utilizes arrested timelines and unequal division to consolidate resources, discarding material to yield just one massive, heavily fortified target. Yet both biological systems flawlessly execute their core requirement, reducing the 46-chromosome genome down to a haploid state of 23. Evolution applied the exact same genetic mechanism to solve 2 opposing biological challenges. The result demonstrates that at the microscopic level, structural form is strictly and entirely dictated by strategic function.